This is screencast number 4 for Chapter 7. I had previously introduced the concept of pesticides as part of industrial agriculture, and that pesticides can be classified into broad spectrum or selective, and further classified as persistent or non-persistent. I had mentioned the pesticide treadmill and how there was no increase in actual agricultural production as a result of using pesticides. And I believe I mentioned that another issue with pesticides is that it can kill beneficial organisms as well as the target organism. So now I want to look at two other issues that are related to pesticides, and those are called biomagnification and bioaccumulation. So we'll look at those on the next slide. First, let's look at the definition. Biology, right? So living things, accumulate, to increase in amount. So bioaccumulation is when pesticides build up in an organism, in one particular organism, over time in the fatty tissues of consumers or predators. So bioaccumulation, build up over time in organisms, one particular organism building up more and more pesticide in its system. A related concept or issue is biomagnification, and that is an increase in the concentration of a chemical as it moves up the food chain or trophic pyramid. So we're going to look next at the example of DDT. DDT bioaccumulates and biomagnifies. So we're starting with DDT in the water. It's present in only a very small quantity, 0.000003 parts per million, which means parts per million molecules of water. Anyway, that's a very low concentration. Then very small organisms called zooplankton ingest this water, and the concentration of DDT then starts accumulating in their cell. And that increases to a concentration of just 0.04, so many more times than it was originally. Then as you move up this theoretical food chain, you have small fish such as minnows, and they feed on the zooplankton. And then as they feed on zooplankton, more and more of that DDT concentrates in their systems, and we're up to 0.5 parts per million. Then we move up to larger fish, such as a needlefish. And then the DDT from those fish accumulates even more. And then we have a magnification up to 2 parts per million. And then finally, we have birds that eat fish, such as ospreys. And the concentration in their tissues can increase as high as 25 parts per million. So the DDT accumulates in the individual's tissues, but then its concentration is magnified as you move up the food chain. And I believe we talked about this in class, or we'll talk about it in class, but the effect on a particular organism may not be direct, like poisoning of that organism. What can happen in the case of the ospreys is the DDT caused their shells to become thinner and more fragile. And they're large birds, so as they walked around on their nest, many of the eggs were broken, which led to a very steep decline in the population. And by the way, DDT is also very persistent. Even though it was banned years ago, it's still around. Okay. Next, we're going to turn to the topic of genetic engineering. So I'll run through some definitions and the goals, and then we'll look at the specific examples and some of the negative possibilities. So first of all, what is genetic engineering or genetic modification? That's when a gene is transferred artificially to another organism to produce desirable traits in the recipient. So before going ahead and looking at what some of the traits might be, let's talk about the genetic modification process itself. This is completely different than what has been done for thousands of years. For thousands of years, humans have chosen which organisms in an agricultural setting, which organisms will mate and produce offspring. And that's artificial selection. That's not genetic modification. So they're seeking to get corn that's larger and has better kernels, or a tomato that ripens faster or better, or is bigger, for example. So that's artificial selection. Genetic modification is very different. In genetic modification, the two organisms are typically not even remotely related to one another. So it's not like you are combining something from a gene from an orange with a gene from a grapefruit. Most typically, it's a gene from a bacteria cell that is injected into a plant or animal cell. So bacteria and plants are very, very different. And it's a specific gene in a laboratory that's withdrawn from the bacterial cell and put into the plant cell beginning at the early stage, the seed stage, or even before that. Anyway, so that's genetic modification. Now, the goal is to produce plants just like the old way, but even faster. So plants that can produce a greater yield. Perhaps it's because they grow faster, perhaps they grow bigger. That would lead to a greater yield. And that would be great because, as you know, we have food insecurity in the United States and globally. So theoretically, the more yield you have, the more people that can be fed. A second goal is to improve food quality. So the example that your textbook gives is very good. It's called golden rice. And in many communities that rely on rice as a major source of their calories, there is a problem in that they lack vitamin A. And the lack of vitamin A in childhood can result in blindness. And so that's obviously a tragic outcome based on just missing this one vitamin. And so the idea here was that golden rice are rice in which they have inserted a gene which allows that rice to be able to produce vitamin A, and that would improve the quality, the health of that food. A third goal is to reduce pesticide use, and this gets kind of complicated. Corn has a pest, like a boring worm, and it competes with humans for corn. And for the last 70 years or so, heavy amounts of pesticides were used on corn crops to compete with this pest, or to eliminate this pest. But that means a lot of pesticide use, and we already talked about how that can be problematic. So the idea here is to insert a gene from a bacteria known as Bacillus thuringiensis, or BT for short, and insert that gene into the corn. So that gene, that particular bacteria, is a natural pesticide, if you will. It's not a chemical, but it exudes something which prevents that pest from taking hold of the corn. A fourth goal is to reduce world hunger by increased food production. So that sort of feeds back to number one here. And of course, increased profits, because the greater the yield, the more profits as well. And there are additional benefits that aren't listed here. For example, a plant, yes, you get greater yield, let's say, but a plant may be made drought tolerant, so they can survive with less water. That would then give us a greater yield. You can have plants that are more able to withstand wind, for example, or salty air, or salty soil, certain minerals in the soil. And so all of these lead to beneficial results. Okay, now let's take a quick look at the concerns about genetically modified organisms. One is the safety for human consumption. So how do we do a study on whether a genetically modified organism that's consumed as food, whether it is safe or produces any negative effects on a human? Well, food studies are incredibly hard to conduct. First of all, genetically modified foods were introduced before doing really a test for safety on human consumption. Secondly, these types of safety issues typically take place, as I think we talked a little bit about in earlier chapters, but things may not show up for decades. It may take decades of exposure to something to be able to see a safety issue. It may also take multiple exposures, so the degree of exposure to a particular item before it's known that something is harmful. So that happened, for example, with cigarette smoking. It took a number of decades before people realized that that was associated with lung cancer and COPD. So as a substitute for testing human consumption, safety for human consumption, which is, again, hard to do. How do you list every food that a person eats for a year, much less 10 years? Animal safety studies are conducted instead to determine whether any ill effects are seen. So some of these studies have conflicting results. And there are various possibilities as to why that is. Sometimes that just happens in science. There's also an issue about whether these studies are done typically through agricultural people who are interested in agriculture and may even be involved with genetically modifying organisms themselves. So the safety for human consumption is still a question that's out there. One example that opponents to GMOs point out is the rise in celiac disease, the number of, and peanut allergies as well, the number of people who can't tolerate wheat or sometimes oats or peanuts or even have an allergic reaction. And they wonder if that is tied to the introduction of GMOs. Okay, second effect is the effect on biodiversity. So you know that biodiversity is very important, and it's very important within one species to have a variety of organisms, and that protects that species, or that population rather, well, both, from becoming adversely affected by any changes in the environment. So, for example, the Irish potato famine was a fungus that affected a number of potatoes, and there was not very much biodiversity in the potato crops in Ireland at the time. So over a period of a couple of years, this lack of variety within a particular food crop did result in the food crop failure, and that's what led a lot of folks from Ireland to immigrate to the United States. So the same kind of effect on biodiversity is of concern if we have genetically modified organisms that are all pretty uniform. They even have kill seeds, which is another issue, but at any rate. So the concern is that if you have a crop that's all the same genetically, there could be a pest or something that is able to destroy those crops. And if you don't have that diversity, then you could lose your food crop. I'll talk more on that in just a minute. Last, I want to mention how to regulate genetically modified organisms has been an issue. We're a little short on time, so what I'm going to do is continue on to another screencast, talking just briefly more about effects on biodiversity and regulation of GMOs.